bienvenida en la Universidad Nacional de Río Cuarto. Nos disponemos a dar comienzo a la charla magistral sobre bioestimulantes vegetales, repensando la nutrición vegetal, a cargo del profesor Patrick du Jardin de la Universidad de Lieja, en Bélgica. Esta conferencia, a la que vamos a dar inicio, es organizada por la Facultad de Ciencias Exactas, Físico-Químicas y Naturales de nuestra Universidad Nacional y el Instituto de Investigación Agrobiotecnológica dependiente del CONICET. Patrick du Jardin es profesor emérito de Fisiología Vegetal en la Universidad de Lieja, Bélgica, y tuvo un rol decisivo en el desarrollo conceptual y regulatorio de los bioestimulantes en todo el mundo, pero especialmente en Europa, donde se lo considera como una voz influyente en la discusión sobre innovación y sostenibilidad en agricultura. Cabe recordar además que el profesor Patrick du Jardin fue declarado profesor extraordinario visitante mediante la resolución de Canal 123/2025. Se encuentran presentes aquí la profesora Marta Dardanelli, secretaria de Ciencia y Técnica de nuestra Universidad Nacional de Río Cuarto, el profesor Sergio Alemano, director del Instituto de Investigaciones Agrobiotecnológicas de nuestra universidad, el profesor Fabricio Casán, docente de nuestra universidad e investigador principal del CONICET, y en el día de hoy nos complace con su visita el profesor Patrick du Jardin de la Universidad de Lieja, Bélgica. También se encuentran en la sala demás autoridades de nuestra universidad y de los distintos institutos, docentes, alumnos, participantes también de esta conferencia, a quienes damos la cordial bienvenida. Cabe mencionar que esta charla, a la que estamos dando inicio, será transmitida en vivo por el canal de YouTube de UniRio TV, el canal de nuestra Universidad Nacional de Río Cuarto, y también en su canal propio. Invitamos a continuación al profesor Fabricio Casán, docente de nuestra universidad e investigador principal del CONICET, para darles la bienvenida a esta charla y agradecemos, por supuesto, a todos nuevamente su presencia.
Bueno, muchas gracias a todos. Voy a hacer la presentación en inglés para formalizar la presentación para Patrick. Thank you, Patrick, to be here today. It's an honor to having you and your knowledge here today. And because we are an agronomic university and biology university, and the biology bounded to the agronomic production, we are really happy to be here today and listen to you. And in the name of the faculty, in the name of the researchers, students, and everybody here today, we give you a warming welcome and have a nice presentation.
Bien. Okay, thank you very much. Yes. May I use this? Yes. Take this out. As a teacher, I used to stand up when I give my class.
Aprovechar para invitar también a quienes están dando por finalizado el acto a descender y dar inicio a esta charla. El profesor, si también desea realizar alguna presentación en particular acerca de su significado. Muy atento. Muchas gracias. Damos comienzo entonces a la charla.
Okay, thank you very much for the invitation. I'm sorry that I don't speak Spanish. I understand some Spanish, which allows me to say I'm not emeritus professor of plant physiology, I'm still professor of plant physiology. And happy to teach my students about the complexity of plants and how this complexity needs to be tackled for agriculture, for bioengineering, and there are so many challenges of agriculture today that call for a better understanding of the physiology of plants. And I think that plant biostimulants probably are a perfect illustration of how plant physiology is important for moving forward with agriculture. So I propose this title, indeed, Plant Biostimulants, Revisiting Plant Nutrition. What are biostimulants? Probably you're a bit familiar with the concept, but I would like to revisit the concept with you and to try to go further in the understanding of the concept. Well, I would say plant biostimulants is some kind of physiological effector. Effector is something that has an action on, okay? It affects. It enhances plant growth, development, metabolism. For me, that would be a way to define, although it's not in the regulatory definitions, but that's probably the way we can understand a biostimulant. And I would like to help you understand better this concept with an analogy with human nutrition. I like lemon juice, especially after a good meal, eating fats, drinking alcohol, always too much alcohol, because alcohol is a poison, so you cannot drink even a small amount is a small amount of poison, okay? It challenges your body, your liver, and you have to protect your body against oxidative attack. You need also to help your body digest the fats, producing bile for the emulsion of fats. And it's quite traditional to have a lemon juice or a lemon sorbet at the end of a fatty meal. Why? But not because lemon juice provides you with calories, although there are sugars there. Okay, it is a source of nutrients. It's an interesting source of potassium, iron, etc. But when I have a lemon juice after a heavy meal, it's not because of the sugars and the potassium. It's because all these bioactive components, which are well described by the scientific literature, and that helps your body producing more antioxidants to stimulate the secretion of bile, to help digestion of fats. So basically, lemon juice is a biostimulant. It is a stimulant of specific functions, physiological functions. And that's the biostimulant. You're interested in specific physiological functions, not of your body, but of the body of the plant. And basically, that's new. That is what is new with biostimulant. Easy to say, not easy to implement in the practice. You have all kinds of biostimulant products. You have non-microbial, microbial products. We will talk about some of them, of course. You can have all kinds of treatments, foliar spray, seed coating, etc. You can apply this on all kinds of crops. And the expected outcome of this is to improve nutrient use efficiency, to enhance the tolerance to abiotic stress, to increase crop quality, and after all, of course, to increase yields, because this is something that will be important for the farmer, and that might be the consequence of a better nutrition, consequence of an enhanced tolerance to abiotic stress. Yield, of course, will be maybe the final output of this. But we will see that the regulatory definitions of biostimulants will pay attention of specific claims, which are the expected outcomes of applying the product. Okay, are biostimulants new? Well, no, certainly not. Certainly not. And I tried to fish out the first article using the word biostimulant in the title. And this paper is from 1990. It is titled "The Use of Organic Biostimulants to Help Low Input Sustainable Agriculture." It's a paper published by an American group from Yale University using mixture of seaweed extracts, humic acids, some vitamins, applying this to different type of plants at the seedling stage, including pine seedlings. It's a forestry department. And they show that you can reduce the supply of nutrients, add this biostimulant, and you have perfect growth of the plant. You can reduce the supply of nutrients and compensate this with the biostimulants. And they explain that this is because the plant is more efficient in the way they use nutrients. And also they show some enhanced drought tolerance of the plant. So basically, the main claims of biostimulant action are already present in this paper. And the biostimulants we talk about are the classical ones, seaweed extract, humic acids, etc. Yes, but at the same time, we also have people trying to understand how this works. And there is a paper there talking about biostimulants, seaweed extract, humic acids. But this is about phytochelates, okay, in plant growth and productivity. So molecules able to complex or to chelate micronutrients, zinc, copper, etc. And they say that, oh, these biostimulants like humic acids, humic and fulvic acids, they are these molecules also able to complex micronutrients and helping the plant better use them. So they might be one of these natural phytochelates. But we have a problem. Inconsistent product performance in the field has been a major factor limiting the application of biostimulants in agriculture and horticulture. This is from 1993. We are 30 years later, more than 30 years later, and we can still say the same. The problem with biostimulants is probably still today inconsistent product performance in the field. Okay, we have to understand that. This comes from a commercial folder from a company. What you see here is the yield impact, positive impact on yield, negative impact of yield. When you use seed coating, you use a biostimulant which is based on humic and fulvic acids. You make seed coating of wheat seeds, and you perform 73 trials with these different varieties, different soil conditions. Okay, but what you see is that you have a huge variation in the impact of the application of the biostimulants. The company is happy of this because they publish this in a folder, and it's a commercial one. But still, for the farmer, of course, it's a bit uneasy. I want to be sure not to be there. But can you tell me in which conditions, which recipient variety will give this and which one will give this? And the problem today is that we hardly know which are the conditions for the performance of these biostimulants. Do we have, years later, after these first pioneering works, do we have some meta-analysis of the effects of plant biostimulants on crop yields? We do have a nice study on non-microbial biostimulants from 2022, and another one from 2018 on microbial biostimulants. All papers talking about yield impact and trying to understand in which conditions do we have a statistical trend to have the highest yield increases, in which can we understand? Of course, there is a bias when you do this kind of analysis, because they take lots of data published in peer-reviewed journals. But there is a bias, of course, that when you publish something, it's because you have a positive effect, usually. It's easier to publish when you have a positive effect than to publish when you have no effect, okay? So we have to be careful when interpreting this data. But this is a summary of the data for the non-microbial biostimulants with an average yield benefit of 17.9%, but as I just said, published means bias towards positive results. But where do we observe the highest benefits? Usually from soil treatments, on vegetable crops, under suboptimal growing conditions, where you have water limitation, limitations in nutrients, low soil organic matter content, etc. So that's maybe the most interesting part. And you can look to the figures. I will not have the time to go to the details, explaining which conditions, like strongly saline conditions, you tend to have highest benefits as compared with non-saline soil conditions, etc. So you have statistical trends. You can, okay, it's a nice paper, But of course, it's not the final conclusion. You cannot say for any crop with any biostimulant you will have higher benefits in drought conditions or saline conditions. It's statistics. Okay. You have also this work on the microbial biostimulants, focusing on the responses of yield, nitrogen use efficiency, phosphorus use efficiency, and the microbial biostimulants were nitrogen fixers and phosphate solubilizers, or their combination. And again, you do have positive effects overall, which are nicely described. Highest benefits with the AMF type mycorrhiza on legume crops, okay, with the application of rhizobia in arid climates, etc. So that's interesting. Okay, I can recommend these papers if you want to know more about it, of course. But is yield response the right metrics? The thing we have to measure when we want to understand biostimulants? I'm not sure. Especially if you read the regulatory definitions. In Europe, we had the first regulation on fertilizing products, including biostimulants. And there we have the definition, which you can read here. Upon biostimulant shall be an EU fertilizing product, the function of which is to stimulate plant nutrition processes independently of the product's nutrient content, with the sole aim of improving one or more of the following characteristics of the plant: nutrient use efficiency, tolerance to abiotic stress, quality traits, or availability of confined nutrients in the soil or rhizosphere. That means that we define the biostimulants not by higher yields, okay, but by specific claims, the specific claims from A to D. This is how we will define biostimulants. And that means that we have to decide using a biostimulant when we have a clear idea about the expected outcome, not only yield at the end, but before that, before yield, what is the expected outcome? You have to build a narrative if you want to choose the right biostimulant. You have to know what you're interested in. And I give a couple of examples here. You have the agricultural target, left column. Seedling establishment. You're interested in early root growth for the acquisition of nutrients, phosphate acquisition by the young maize seedlings, for instance. Then you can describe which kind of biostimulant action you're interested in, which is humic acids has the capacity to promote root growth, definitely. And you have published evidence in the literature. You need to have published evidence in the literature to be able to find this. And the claim, which is one of those claims defined by the regulation, is improved nutrient use efficiency. You need to be able to connect these agricultural targets, these biostimulant actions, and the final claims. Another example, I will not take all of them, but another example which might be interesting to share is you might be interested in delay the pod shattering in oilseed rape or in soybean. In Belgium we were more interested in oilseed rape than in soybean, but the opposite is true for you. And we have published evidence that in seaweed extracts you do have hormonal activity, oxinic activity, which is able to retard the opening of the pod. Okay? And this will translate into quality traits, one of these quality traits, and also in yields. Again, you have to build this narrative, okay, if you want to address the biostimulants, to be able to define a proper use of these biostimulants. Other examples, I'm willing to share my presentation if you want the details, but I like to quote the Stoic philosopher Seneca. You know the Stoic philosopher Seneca? Of course, everyone knows the philosopher. And I will not quote Seneca in Latin, but take the English translation. He said, if one does not know to which port you're sailing, no wind is favorable. Okay, if you want to evaluate your biostimulant, you have to know what you're interested in, which traits, for what reason. Otherwise, you don't know how to qualify the biostimulant and to judge, to evaluate the efficacy of the biostimulant. Okay, so we have the claims, which are the regulatory basis of the definition. But how to validate the claims? If we have a regulation, we have to provide some indication to the farmer that this product does fulfill the condition, the claims. And we have in the European regulation, we have regulatory provisions there to validate the functional claims. And the principle is to ask the company to have data sets, complying with standards. Standards define which kind of protocols should be used, statistical design of the protocols. If you want to demonstrate drought tolerance, okay, what is drought? How to measure drought? You have standards, okay? And the data sets, the data packages are provided by the companies to a body. We call this a notified body. It's an organization appointed by the European Commission, let's say, which will check, make the compliance check. Okay, we have the data for this, for that, for this, for that, for this, for that. And this, it does comply with the protocols defined by the standards. So the idea is to have what we call a compliance check. This is the way to put a product on the market. You need data, and the data need to satisfy the requirements of the standards. And this is controlled by some authority. This is the principle. In practice, we do have EU harmonized standards, which were defined by a technical committee of the European Committee for Standardization. So we have a very formal way of defining the standards. And at the end, for the end user information, you will have the CE mark. It's very popular in Europe, of course, but in refrigerator, on toys, you have the CE mark, which means that, okay, it does comply with the standards that we have defined for the safety of the product and also for the efficacy of the product in some cases. Okay, and on the biostimulant product, on the package, you will see the CE mark if all the conditions, of course, were fulfilled. So that's the idea. So we have to be clear, as I mentioned, about what we target. What are the targeted agricultural function? What are the plant traits we talk about? If we have plant traits, that means that we have genes somewhere controlling the traits, okay? And then we have tools for changing gene activity. Many tools are genetic tools, classical breeding, but CRISPR-Cas, gene editing technology, GM technology. But more and more we are now thinking about what we can call epigenetic tools. Okay, we do not act on the genes, but we act on the expression of the genes. With plant growth regulators since years, and now more and more with plant biostimulants. So I think we have to see the contribution, the added value of these plant biostimulants in that way. We are moving to more and more epigenetics in plant science, which will allow us to act on the expression of the genome, to understand the basis of this epigenetic mechanism. You apply a seaweed extract at an early phenological stage, and weeks later you have an impact on yield. So the amounts that you add are very low in most cases. Okay, what are the mechanisms of, let's say, cellular memory allowing this effect to be achieved? We have to understand that. But we have probably more and more to understand about plant traits. I give an example here, which I like very much, of Jonathan Lynch from Pennsylvania State University, working on maize, acquisition of water, acquisition of nitrogen, and defining root ideotype. How should the root system look like to have the higher performance for acquisition of water and nitrogen? And maize is complex because you have roots of different origins with different colors here, I will not comment all these routes. And if you want to have the optimal exploration of the soil for the acquisition of water and nitrogen, for phosphorus it will be different. That's a problem, okay? But you will have optimal angle of insertion of the roots, optimal number of root branching. So you have many individual traits that will at the end contribute to this optimal architecture of the root system, okay? And he comes with an interesting concept, the concept of phene. We talk about genes, component of the genome, making the genotype, okay? But we have to think about phenes, which are these individual traits that will contribute to the phenome and to the phenotype. And that's probably, for me, the biggest challenge in plant physiology, in plant breeding, in the use of biostimulants, is to dissect the phenotype into these phenes so that we can target specific phenes contributing to specific phenomes that we're interested in. And Jonathan Lynch talks about this phenome bottleneck, or ability to understand and manipulate the plant genome has far outstripped our understanding of the plant phenome. And that's indeed very complex. You need modelization, but you also need to identify then those biostimulants which will be able to impact specific phenes. Okay, that's the next challenge, of course. And that's one thing we did a couple of years ago with my collaborator Pierre Delaplace. We studied the impact of, this is just an example I want to give. This is a Brachypodium distachyon. Do you know this model grass? You have Arabidopsis, which is very popular. Brachypodium is a nice grass model. And we grow these plants, you see in the squared Petri dish, in the nearly vertical position. And we have a small Petri dish in the middle where we can grow bacteria there, okay? But the bacteria are isolated from the plant. The only way for these bacteria to impact the plant is via the production of volatile compounds that are released in the atmosphere of the box, okay? And what you see after 10 days, the control, okay? And this is the plant with the exposed to these volatile compounds. And you see that the pattern of branching is very affected. So you have the capacity of these volatile compounds to dramatically influence the way root grows, the way they branch, etc. So that means that if you want to understand the action of biostimulants, of course, you need these kind of things. Okay, what kind of phenes we can act on? Does that mean that it will be a good biostimulant at the end? Of course not. Of course not. Because it's not because you have more branched roots that it will contribute to higher nitrogen use efficiency, etc. It's not enough. In my lab, I used to talk with my students. We talk here about bioactivity. We show here bioactivity. There is a biological activity of the volatiles, but we cannot say that there is any biostimulation potential because we need to show that the claim is indeed satisfied. Okay, so we need to pay more attention to the traits. Probably we also need to have more clearly defined products. That was a presentation made by a person from a company a couple of years ago. And I agree that probably the future of biostimulants is to better understand the impact on the specific traits, but also to have more defined products in terms of structure, in terms of function. Many biostimulants today are a mixture of different things. Amino acids, humic acids, plus some vitamins, and micronutrients might be helpful. Okay, it's very difficult to dissect the analysis of the effect of these complex biostimulants. Probably we have to go to more detailed composition and modes of action. Because the difficulty in the regulation today is that you have to demonstrate the claim, but you don't have to demonstrate the exact mode of action of the product. Something you need to do for placing a pesticide on the market. You need to know the physiological target, the receptor, etc. It is not mandatory in the regulation on biostimulants to be very clear about the mechanism of action. But certainly we need to move to that direction. And we have more advanced technologies appearing now, like synthetic peptides. like microRNAs, artificial microRNAs, which are regulators of the expression of genes, you have people now working on this application of exogenous small RNA, regulatory RNAs, which will be able to modify the expression of genes and to deliver some biostimulant effect. On the completely other side, I was visiting the CIAT research center in Cali last year, exactly 1 year ago, and I met people, including Dr. Miriam Pulleman there, who introduced me farmers making their own complex bio-based fertilizers. They talk about bioles or microorganismos de la montaña, a complex mixture of things, fermentation products on the farm using crop residues. Okay, it's Boccacci is also a name which you're familiar with, certainly. But it looks really interesting. If you look how people do that, the confidence they have in what they do on their farm, I'm a bit impressed. So I think, and the message I would like to deliver to people from academia is, we should not only think about products to place on the market with companies, but also to help farmers to develop maybe their own solutions on the farm. But of course, there are many challenges there to standardize maybe the product, the efficacy, to show the safety of the product, absence of contaminants, etc. So there are certainly many challenges, but I think this is an avenue quite interesting to explore. And these complex products, bioles, of course they supply nutrients, they have some protective effects against diseases, but they also have some biostimulant effects, like containing humic acids, etc. So a completely different avenue. Okay, now I would like to spend some minutes with you to reflect on how we could make plant biostimulants more effective and what should we pay attention to. Interactions with the receiving environment, application modes and timing, the manufacturing of the products. And of course, this is not a limiting list. But I was interested in discovering some works. We know very well humic and fulvic acids. There is extensive literature of what you can do by applying these products to plants. But root exudates matter. So roots, they secrete many molecules, and it seems that there is some cross-talking of the root and the soil containing the humic acids in the way that some of these exudates, the acids released, the organic acids released by the exudates, they might be important for changing the structure of the humic acids. They might help release some hormone-like components which are entrapped into these complex structures. So there is evidence that the activity of the root affects the activity and effectivity of the humic acids. So there is a clear interaction via the root exudates, and exudates are also especially important for the dialogue with soil bacteria. There is extensive genetic variation in the composition of the exudates. There are quite nice work in Arabidopsis, etc., showing that you can have really changes in the exudates by specific mutations in specific transporters, and that will affect the interaction with microbes. Another thing which is important is also the commercial source. Not any seaweed extract is the same. And if we take the same source, the brown algae Ascophyllum nodosum, you can take different commercial sources, and testing this on maize seedlings after 10 days in this work. And then you measure many things, many growth parameters, and you make this principal component analysis, and you position there the different extracts which are numbered there from the different sources. And you see that you have quite important variation. It's the same Ascophyllum nodosum, but at the end, the effects are quite different. There is one laminaria introduced there. Some of the Ascophyllum nodosum are closer to laminaria than they are to other Ascophyllum nodosum. Okay, so the way you prepare the seaweed extract is important, definitely, for the activity. The application mode is also important. The same seaweed extract was used in these two papers. The species are different, grapevine on the left, sweet orange on the right. Drought tolerance is of interest in both papers, and conclusion is that, okay, foliar application is better on the left, soil drenching application is better on the right. Okay, it's the same extract, it's not the same plant, okay, which matters, of course, but sometimes people ask, okay, is it better to apply on foliar, as foliar application or soil drenching? It's impossible to generalize. It's impossible to generalize. That's the message. Timing of application, of course, is important, especially for stress tolerance. And I would propose to think about how a product might improve stress tolerance by distinguishing those biostimulants which would act before the stress, prepare the plant, okay, in a way. We call them primers. We have the rescuers, which help the plant during the stress. And then we have the restorers, those which help the recovery after the stress. And if you look closely to the literature, it becomes clear that some biostimulants might be more a primer, another a restorer. You have to understand, try to understand what is the optimal timing of application as compared with the stress episode. Which means probably that we have to better monitor stress as experienced by the crop over the season in the different parts of the field. We have probably to expect some benefit from the association of what we call precision agriculture and the application of biostimulants. You have to try to better monitor stress in the field so that you can also optimize the application. Easy to say, not easy to do, and in order to define these conditions, a good way, this is from my university, so from my faculty, we have what we call an ecotron. I come from Jean Blou, which is the faculty of bioengineering. And we can grow plants in real soils, okay, that we take from the fields in very controlled environments. And this allows us to apply a stressor at a very specific time, okay, and to apply, of course, the biostimulant at a specific time also. So that's one way, of course, to try to understand this. And to define what might be, and for me this is probably one of the most exciting things with biostimulants, is that you can prime the plant, you can prepare the plant to the later stress conditions. And this priming concept, I took one of the first papers there on plants in 2005, and this is a Northern blot experiment, and what you see here is that you have the transcripts which are analyzed for different genes responsive to salicylic acid, responsive to abscisic acid. And you have here a priming agent. The priming agent is a molecule which you apply before the stress. And the stress is application of 300 micromolar sodium chloride. Okay? And you have the control. You have here the plant which is treated with the priming agent, beta-aminobutyric acid, and this is horse post-treatment. It's a time after the stress. And what you do see here, so the BABA was applied 1 day before stress, okay, and this is the kinetics of the induction of the stress responsive gene. You see in the control here, but when you prime the system, the response is quicker and higher. And this is true for different genes which are responsive to stress conditions. So that's one of the first evidence of what we call priming. The amplitude of the response is higher. It starts earlier. You can have different behaviors there. But that's something really important. This concept of priming is, in my view, something important. You have the non-primed state. When you have abiotic stress, you have the normal tolerance. The idea is to have a priming-inducing stimulus so that you have a primed cell which, when challenged by the abiotic stress, will show an enhanced tolerance. That's the concept. And priming is the potentiation of stress response after it has been experienced. It has experienced some stimulus. And the very important thing here is that the priming stimulus can be completely different than the stressor. Okay? We know what we call cold acclimation. Cold acclimation is one priming mechanism. Okay? Some cold before the stress prepares the plant to have more cold after. Okay? It's plant physiologists know that well. But what's interesting is that you can have a chemical priming. Some molecules are able to prime the system for a stress which is completely unrelated with the priming stimulus. Okay? Priming is also important because the primed state is a way the plant is in a state of alert. Okay? It's getting ready to respond, but it does not respond. Which means that the cost of the priming state is very low. There is no reorientation of the genes which are expressed, the proteins produced. Okay? The system is put into a state of alert. But the cost is very limited. But when the system is challenged by the stress, okay, then you have a better response. Fitness is maintained relatively high as compared with the unprimed system where you will have the drop in the performance. And the benefit in stress situations is there. The cost when no stress is there is limited as compared with the benefit. I think this is important. But this needs good work of plant physiology, of course. And plant care is more than plant cure. That would also be a message I would like to deliver for the end of my talk. The source of inspiration is how we define human health. Okay, we started from human nutrition, now we are arriving to human health. If you see the definition of human health from the World Health Organization, it's not only the absence of disease. Health is more than the absence of disease. It's a state of complete physical, mental and social well-being. Okay? I would propose that we move to this positive concept, enlarged concept of plant health. We start from a really negative health concept in plants, which is the absence of disease. If you look to the regulation in Europe, but elsewhere, what do you do to improve plant health? To combat the diseases and pests, you have quarantine measures, preventing the entry on a territory of a disease. But that's all, I would say. Okay? I would propose to move to this more positive definition of plant health, which is the capacity to carry out its physiological functions to the best of its genetic potential. That's a positive way of defining plant health, which in a way is inspired by this human health as defined by World Health Organization. Which means that if you want to improve plant health, of course plant protection products are important to combat diseases, but biostimulant products will also probably be important to increase plant health because it will increase the capacity of the plant to carry out its physiological functions to the best of its genetic potential, even in adverse conditions when you have stress conditions. And I'm ambitious. I would like to propose a definition of plant nutrition then. Plant nutrition is the acquisition of inorganic nutrients, of course NPK and everything you know, but might be also the acquisition of organic substances by plants, allowing them to carry out their physiological functions, supporting growth, development and health. Plant nutrition is more than just acquisition of the building blocks for growing. It might be the acquisition of what helps you to deliver all your functions in a wide range of environments. And this will be my conclusion. This is from the Ayurveda tradition. When the diet is wrong, medicine, there's a misspelling there, medicine is of no use. When the diet is right, medicine is of no need. I would propose to adapt this to crop nutrition, saying that crop nutrition is crop protection. When crop nutrition is wrong, plant protection is of no use. When crop nutrition is right, plant protection is of no need. Probably we can go that far. I'm a bit provocative, but I think that we might go into that direction. And I thank you for your attention, and I'm willing to answer any questions, of course. Thank you.
Questions? Yes.
Hi, Patrick. Thank you for your presentation. I have one or two questions. One of these is seaweed extract, according to your experience, is the more biostimulant using in agriculture? And the second one is, you think that the potassium phosphate or zinc phosphate is a biostimulant or not? In Spanish is fosfito is okay. Thank you.
Thank you. Thank you for the important question. First, seaweed extracts, together with humic acids and with protein hydrolysates, if you take those three, you have most of applications today. And these are really three important categories on the market today. But you're right, seaweed extract. I'm not sure seaweed extract is the biggest one. They are quite similar in terms of adoption, I would say. So phosphito, okay, does have biostimulant effects. That's clear from the literature. That's clear. But the first application and the first reason for placing these products on the market are for biocontrol. So for combating diseases, for getting the plant also more effective in combating diseases. The story about phosphate, maybe anecdotal story, is that the European Commission wanted to avoid that there is some parallel way of placing phosphate on the market as a biostimulant, because it's a fast track, it's less expensive, etc. But that the product would still be used for disease prevention. Yeah, Phytophthora or whatever. And the position of the European Commission was quite strong, excluding phosphate explicitly in the regulation, which was very strong as a standpoint, but that's the answer of the European regulation, which I will not give this as the example to be followed everywhere, but we have to be aware indeed that we can have both effects on disease prevention and some biostimulation effect. What I would not say, like some people say it also, is that, okay, it can be a source of phosphorus. Only in very special situations where you have some microbial activity allowing the oxidation of, etc., you might have some effect, really nutritional effect of phosphate. But I think for that we have to be clear. No, it's not a supplier of phosphorus. But yes, you can have some biostimulation effect that was shown in the literature. Welcome.
Yes, wait. Yo creo que voy más rápido.
Hi.
Hi.
Patrick du Jardin.
Yes, that's me.
I'm Laura Barrio Nuevo, an agronomic engineer. I'm ex-CONICET. I work here for 2 years, and I have a job in Mendoza. I'm making potato seeds, native potato seeds without virus for selling to Europe, organic. Where we have almost 40 varieties of natives. We have one greenhouse in Tupungato, Mendoza, and one greenhouse in Inta, Mendoza. And in Tupungato, we have had alternaria problems, but we were using organic fertilizer once a week since seedlings to harvest. We have like the 11 varieties had alternaria, but because of the fertilization, some of them don't. And some of them are 5, and some of them are 0. I forgot the question, I'm sorry. Because I hadn't spoken English since 2014. I'm sorry.
But I understand your English at least. Yeah, but you see that you see evidence that nutrition and disease control are really tightly connected. We know also that many we can take copper sulfate, which can be both a supplier of nutrients, of course, but also helps to control diseases in vineyards and other species. So the connection between nutrition and plant protection is really important and probably unexplored. With biostimulants, we have, I think, a way to develop this idea, but including with, let's say, classical fertilizers. We know that we have an interaction with disease protection, clearly. Yeah.
Thank you, Dr. Dujardin, for your excellent conference, the first. And then is a big question. What is the difference, the effects between quatine seed application or foliar application of a biostimulant?
Well, it's very difficult to answer, as I said. Otherwise, I would say the opposite of what I said before. It's very difficult to generalize, okay? But of course, you have to think about what is the expected outcome. In some cases, I was discussing with people who took me with his car from Cordoba to hear about the importance of seedling establishment in some with some places with cold temperatures in spring, etc. So where you can help the plant develop with phytoregulators, mixture of hormones, okay? And the funny thing is that these people work with Stoller, and they have a product they call Stimulate. But Stimulate is not a biostimulant, it's a mixture of 3 hormones. But you do have an impact, you're interested there in, okay, let's help the plant develop in spring under cold temperatures, and we have evidence that, okay, we do have effects from phytohormones, but seaweed extracts also have phytohormones. So you can also have similar effects, I would say, with some biostimulants at least. So you target a specific context. I think it's very important to think about phenology, the development of the plant, and when you expect some effect when applying at this stage for delivering this quality trait or this yield component of whatever. In some case, foliar application will be key. to support maybe the stay green effects for filling of the grains in cereals, etc. We know that we have lots to be learned from the literature on plant growth regulators also. Because many plant biostimulants definitely they play with hormones, not necessarily because they contain hormones, but they interfere with the biosynthetic pathways of hormones, etc. So we have evidence for that. So in some cases, foliar application, if you're interested in the stay green effect for drought tolerance in some cereals at a late phenological stage, okay, foliar application is the way. At other phenological stages for other agricultural outputs, the seed coating will be the answer. Of course, what's interesting is to see how long can be the effect when you have seed coating, how long you have the effects throughout the development of the plant. And that's for me a very exciting field for physiologists and molecular biologists to think about the memory of cells. I was talking about epigenetics. How can a prime state be passed along the mitotic generations? Not to talk about passing through meiosis. We have some evidence also of epigenetic mechanisms passing through meiosis. So this is a very complex field of biology, but I believe this is really relevant for understanding biostimulants.
Another further question? Yes.
Thank you for your presentation. I have one question. When we apply biostimulant, we are going to have the response about the minimum law. When we apply biostimulant, do you recommend to apply different source of nutrition to have better response of the biostimulant? And what is the best source of nutrition to apply with biostimulants?
Again, sorry, but I don't think we can generalize. We should think about specific nutrients, whether you talk about micronutrients like zinc, copper, or whether you talk about macronutrients like nitrogen. Lots still to be done to improve nitrogen use efficiency in plants. So which biostimulant solution will help you to deliver the best effect, and also which combination you have to think about is really something that is tightly dependent on the agronomic context. Definitely, yeah.
Okay, and different kind of source of nutrition maybe interact with the biostimulant. For example, if you have with lowest pH or something like this, maybe can interact and we don't have the best response.
Yes.
One or the other.
Yes, you're right. Yes, pH, of course, is very important for activity of many biostimulants, including microbial biostimulants. And of course, I understand maybe better your question, the choice that you can make of the fertilizer, because they impact the pH differently, depending on the source, might also impact, of course, the activity of the biostimulant. That's possibility. I don't have examples in mind, but from a theoretical point of view, certainly. I think we have really to think together. Plant nutrition, the supply of nutrients, disease prevention, stress tolerance, trying to bridge all this to optimize nutrition in the broadest sense, contributing to what I call plant health, also in an expanded understanding of the concept.
Thank
you. Another question? How to differentiate the effects of the non-microbial and the microbial effects a long time? Because you are providing, when you use microbial biostimulants, you are providing living cells, and probably the effects are longer in comparison with some biostimulants in which you expect to see a bigger effect at the beginning and then to preserve the effect along the time. But maybe we think in different way, the microbial and non-microbial biostimulant, if we follow your recommendations. What do you think about that?
Well, I think it's still with microbial biostimulants. It is challenging to have the establishment of these bacteria into the rhizosphere, etc. Again, within what we call microbial biostimulants, especially in the European regulation today, we have those symbiotic rhizobia, etc., even mycorrhiza. Okay, so if you have establishment, if you have the nodules there, etc., okay, then you have a long-lasting effects. But many microbial biostimulants are not nodule forming. They are in the rhizosphere, and it's clearly not easy to know when they will establish for a long term or not. You're right that you might expect a longer duration if you do have establishment in the rhizosphere, but it's still challenging. And the point is that even with non-microbial biostimulants applied early, you can have effects late in the development.
I'm thinking in foliar inoculants or microbial biostimulants in which you see an effect, but normally the companies or the effects related to those microbes are not evaluated a long time. You can see a good effect at the beginning and you don't know why. But the thing is probably the way to analyze the colonization or wherever must be important to define the action, the mode of action of the microbial biostimulants.
We definitely need microbiologists.
Okay.
Fabricio.
Another question? If you don't have one more question, I would like to thank you, Patrick, again, one more time, to be here today.
I thank you. I thank you for the invitation. I'm not sure I did say it in the beginning of my talk. I really warmly thank you for organizing my stay also here. Fabricio, it's a real pleasure to know you, to have a nice assistance, which shows that you're interested by biostimulants, and definitely we need more science in the field. More science, but also more agronomy. Agronomy is a science, okay. Biological sciences, agronomy, ecology.
I will move to Spanish to few words.
Yeah.
En Argentina, desde hace 2 años, la normativa que regula el uso de biológicos en agricultura considera particularmente una categoría que es la de bioestimulantes. O sea que eso nos pone a nosotros en la necesidad de entender mejor cómo funcionan, y si bien es una cuestión normativa, yo creo que ya al existir una definición y las características que determinan esa definición, tenemos que entenderla y obviamente estudiarla. Tenemos mucho background en la microbiología asociada a los bioestimulantes, pero en otros tipos de bioestimulantes creo que tenemos que avanzar mucho, así que por eso considero una buena oportunidad que Patrick nos visitara y nos trajera este mundo de bioestimulantes de la Universidad de Río Cuarto, y nosotros estamos más felices que él de que él haya venido a la universidad. We are more happy than you that you were here today, and thank you one more time. Okay. Gracias a todos ustedes por la